Welcome. In this session, I'm going to discuss about gymnosperms and angiosperms. So let's see characters of gymnosperms. Right. So in the last sessions, we understood that the gymnosperms and angiosperms are the only plants who produce seeds. Okay. So here, gymnosperms are commonly called naked seeded plants. Naked seeded plants. So why they are called naked seeded plants? So here the seeds are not covered by fruit. So here there is no fruit developed because ovary is absent. So we know that ovary, I just mentioned, so ovary is absent, but ovules are present. So in case of gymnosperms, ovary is absent, ovules are present. And we know after fertilization, the ovary actually develops into fruit, whereas the ovules will develop into seed, will develop into seed. As here there is no ovary, okay, the ovules are seen to outside, but in case of angiosperms we see that these seeds are covered by the fruit. So there in angiosperms the ovules are not naked, but in this gymnosperms, in this group of plants, okay, the ovules are, okay, seen to outside. So because of that they are called naked seeded plants. Right. The next one here, coming to the range, the plant size. So it includes shrubs. So there are no herbs in case of gymnosperms. So it will be shrubs. Then we'll also see medium-sized trees to tall trees. Okay, so this will be its a habit. Okay, shrubs, medium-sized trees to tall trees. An example for this tall tree, I'll just mention, it's a giant redwood tree. So this is a very tall tree species. Its scientific name is Sequoia dendron, okay, giganteum. So it's called Sequoia dendron giganteum is its a scientific name. So of course, in short, I'll just mention as a sequoia. So its complete name is Sequoia dendron giganteum. So this is a very tall tree species. Is a tall tree species. Okay. The next one. So here in some genera there is an association of fungus with the roots. Okay. So some species of gymnosperms have an association with the fungus. Okay, so and they result in the formation of mycorrhizae. So in some species mycorrhizae, okay, is seen, right? So we know the meaning of mycorrhiza, the symbiotic association between the roots of plants plus, okay, the fungi. So when such an association we see, we call it a mycorrhizae. Okay, so such mycorrhizal associations are seen in case of some genera of gymnosperms. Not all will show this mycorrhiza. All species will not show. Only few genera will show. Like in case of pinus. So in pinus we see such mycorrhizal association. Okay, and whereas in some other species we find special roots called coralloid roots. So these are okay associated. So here coralloid roots, which means where the roots of these plants, okay, so roots of these plants plus okay cyanobacteria. So here they are associated with the cyanobacteria, the blue-green algae. So coralloid roots are an association of roots plus the cyanobacteria. So the cyanobacteria will inhabit these roots of the plants, okay, where it actually help in fixing nitrogen. So this association helps in fixing nitrogen. So helps in fixing nitrogen. In such coralloid roots, we can see in case of cycas. So example, in case of cycas, we will see these coralloid roots. In case of pinus, we can see this mycorrhizae association. Okay, then coming to stems. So here, in some cases, the stems are unbranched. So like we see in case of cycas. So stems are unbranched in case of cycas, and so the stems are branched. So in some species, the stems are branched, like in case of Cedrus, Pinus, etc. So in them, the stems are branched. Then coming to the leaves here also may be simple or compound. So the leaves may be simple or compound. So we know the simple leaf, if the leaf lamina is whole, it is simple, and if it divides to form leaflets, then the condition is called compound leaf. Okay, so the leaves in these gymnosperms, they may be simple or compound leaves. Then okay, in case of cycas, so we see pinnately compound leaves are present. So we see pinnately compound leaves in case of cycas. But these leaves are present only for few years. Okay, but only for few years we see the presence of these pinnately compound leaves. Then here coming to the leaves in gymnosperms, they are well adapted for different environmental conditions. Okay, so here the leaves in gymnosperms are adopted for withstanding high temperatures, then humidity. So they are capable of withstanding high temperatures, humidity, and also wind. Okay, so these extreme conditions, the leaves are very much well adapted. Okay, and in a group of plants of gymnosperms, which are commonly called conifers. So conifers are gymnosperms, and these conifers, they have needle-like leaves. Have needle-like leaves. Conifers have needle-like leaves, and the presence of these needle-like leaves actually it helps in reducing the transpiration, that is the water loss. Okay, so here these needle-like leaves actually reduce surface area, so they reduce the surface area. So here if we see in case of a leaf that has a broad lamina in this way, it has more surface area, so hence there is more presence of stomata and more loss of water. And if we see in case of a leaf so that has less surface area, so say suppose this is a leaf like sorry, needle like leaf. Okay, so here this is a needle like leaf, and in this case you can see the surface area is very less, and hence the occurrence of stomata will be less, and the loss of water will be also less. So here due to presence of these needle like leaves which reduce the surface area, so hence reduce water loss. So that reduction in the surface area will help in reducing water loss. Now, besides having these needle-like leaves, they also have, okay, also have thick cuticles, and sunken stomatas. So they have thick cuticle and sunken stomata. Okay, so here the thick cuticles, it's a waxy layer. We know cuticle is a waxy layer that will be present on the surface of leaf. So it is present on the surface of the leaf. Okay, the thicker the cuticle, the more is it helps in preventing water loss. So here upper and lower surfaces of the leaf, the upper epidermis, and say this is the lower epidermis, and here okay covering is the cuticle. So this is cuticle. So we find the cuticle both on upper and lower surfaces. Okay, so if cuticle is thinner, then water loss will be okay more, and if the cuticle is thicker, then water loss is reduced. So that's why to reduce water loss in case of these plants, okay, in gymnosperms you may find there will be thick cuticle. So this thick cuticle will help in preventing water loss. And they also have sunken stomata. So we know that the upper epidermis will have stomata here. Okay, so but that is present on the surface. But here, as they told, sunken stomata, so it forms pits. So it is seen something like this, epidermis, and here you may find the stomata. So you can see here okay, so this is the epidermis, okay, covered by cuticle here. Okay, so this is cuticle. Okay, and you can see these stomata are present in a pit, and which will be usually covered by some hair like structures. So in this is a okay mechanism that again help in preventing water loss. Okay, so you can see these are what we call sunken stomata. So we call them sunken stomata. So again, the thick cuticle and sunken stomata, both these conditions help to reduce water loss. Reduce water loss. Okay, then so these are some morphological characters. Coming to reproduction, gymnosperms, all gymnosperms are heterosporous. So in earlier case we have seen some as homosporous and some as heterosporous, and I told you that heterosporous is a precursor okay to formation of the seeds. Of course, there the retaining of the female gametophyte, okay, on the sporophyte, and further development of it is a precursor, I told you. So here in case of gymnosperms, okay, so these are all heterosporous, are heterosporous. We know heterosporous, which means they are going to produce two types of spores. Okay, so two types of spores we call microspores, and another one called the megaspores. Okay, so microspores and megaspores, and these spores will be produced within the sporangia, we know it already, and these sporangia will be present on leaves, and we call them sporophylls, and these sporophylls will be spirally, I mean, arranged. So they are spirally arranged on a long axis. So that's what, like how we have seen in case of earlier group of pteridophytes. Okay, so on long axis, and they are going to form cones. So they are going to form cones. So here, so we may see like this. Okay, so this is how we will see. Okay, a cone, right? So these are all sporophylls, or sporophylls. Okay, right? So these microspores and megaspores, they will develop, okay, within the sporangia. Okay, and these sporangia are present on sporophylls, and they are arranged spirally on a long axis, and that result in the formation of lax or compact cones. So result in the formation of lact or compact cones, or we can also call them strobili. So strobilus singular, strobili is a plural. Okay, now the cones bearing microsporophylls. So cones having microsporophylls. Okay, so these microsporophylls will have the sporangia that produce microspores. Okay, so the cones having microsporophylls, the microsporophylls will have microsporangia, having microsporophylls and microsporangia, right? So they are called the microsporangiate, microsporangiate, also called the male cones. So here we find male cones and female cones separately. Okay, and the cones having the microsporophylls and microsporangia are called microsporangiate, or okay, the male cones or male strobili. Then, right, so of course we know these microsporangia is the one going to produce within the microsporangia, right, will be the microspores. So here the microspores produced within the microsporangia. So they are produced within microsporangia. Next, the cones having megasporophylls. The cones having megasporophylls and megasporangia. So I can also call these megasporangia as ovules. Okay, so the cones having megasporophylls, okay, and the megasporangia, right? So they are called the megasporangiate, also called the female cones, right? So here, say suppose we have these microsporophylls. These microsporophylls will have microsporangia. Inside the microsporangia there will be development of microspores. And in case of females, so the megasporophylls, and in the megasporophylls we find megasporangia, so which we call them also as ovules, and inside that there will be development of these megaspores. Okay, so here I'll just shortly give you how the cone looks like in a longitudinal section. So here we know these microspores, they are going to develop into the male gametophyte. So already I think in previous sessions I might have mentioned that pollen grain, okay, pollen grain is the male gametophyte, the male gametophyte. Okay, then the embryo sac is the female gametophyte. The pollen grain is male gametophyte, and embryo sac is the female gametophyte. So these microspores are going to develop into the pollen grains. They're going to develop into pollen grains. Okay, so which will be the male gametophyte. Okay, and this male gametophyte is highly reduced, and it is limited with few cells. So it has a limited number of cells. So the pollen grain uh, will be with a limited number of cells, but in lower group of plants we have seen that the gametophyte will be multicellular, independent, okay, and capable of performing photosynthesis. But as it has come to higher group of plants, this is not at all seen. The gametophytes will not be independent, okay, they will be not, okay, multicellular, it will be few celled, okay, and then they will be not again performing photosynthesis, and they will be dependent on sporophytes. So this is what we see as we come to higher group of plants. So this pollen grain is a male gametophyte, and it is limited, it has a limited number of cells. And coming to megaspores, we know they are going to develop into okay uh, the embryo sac, that will be the female gametophyte. Okay, so the microspores, which give rise to pollen grains, and megaspores to embryo sac. Okay, so here, like the next one, the male and female cones may be present on the same plant. So the male cones and the female cones, okay, on same plant. So like in case of pinus. So it means that pinus is a monoecious plant. So it is a monoecious plant. Okay. And in some cases, I mean in some other species, the male cones and female cones, okay, present on different plants. They are present on different plants. Okay, so this condition we can see in case of cycas. Okay, so example, cycas. So hence cycas is a dioecious plant. So dioecious plant. Okay, So the male cones, female cones on same plant maybe, and in some species the male cones and female cones will be present on different plants. Okay, and let's see how these cones will be seen, the male cone and female cones, how they appear in longitudinal sections. So here I told you that the sporophylls, the microsporophylls or megasporophylls, they are arranged, okay, spirally on an axis. So you can see here. So this is the sporophyll with the sporangia. Okay, this is how it looks like in a longitudinal section. So these are microsporophylls. So are microsporophylls. Okay, and these are the sporangia, that is microsporangia. Okay, so all these are microsporangia, and inside this microsporangia we know the microspores will develop, and those microspores, okay, will develop finally into pollen grains. Okay, so you can see the microspores developing inside. And coming to okay, so this is the male cone, so in case of male cone, and coming to the female cone. Okay, so on this female cone, so these are megasporophylls. Okay, so here we find megasporangia. So you can see here the megasporangia. So it's seen, okay, in somewhat like this. So these are all called megasporophylls. So these are all the megasporophylls, and okay, so here is the megasporangia. So this is the megasporangia, or that's what I mentioned it, ovule. Okay, and right, so inside that there will be the megaspores which will develop into embryo sac. So this is in case of the female cone, will be seen in case of female cone. Okay, we'll see especially in case of female cone. Okay, so how the ovule and the megaspores. Okay, so I'll just, okay, I'll take the megasporophyll, and here is the presence of ovule. Okay, so this is how we see. So this is a megasporophyll, and this is the ovule. So here we'll see that ovule. So this ovule is covered by integuments. So it's covered by integuments, and it has opening, what we call it, the micropyle, has opening micropyle. And here inside this is the nucellus tissue. So there is a nucellus tissue inside the ovule. So one of the cell of the nucellus tissue will get differentiated to form the megaspore mother cell, will form megaspore mother cell. This is diploid, and this megaspore mother cell will undergo meiosis and produces 4 megaspores. So it produces 4, okay, megaspores. So here 1, 2, 3, 4 megaspores are produced. Okay, so all these are megaspores. So out of these megaspores, out of 4 megaspores, okay, 3 megaspores degenerate. So 3 megaspores degenerate, and it will be left with only 1 functional megaspore. Will be left with 1 functional megaspore. Okay, so out of this, okay, all these will be lost, and it will be left with only 1 megaspore, and this 1 megaspore will develop into the female gametophyte. So here we can see this is the female gametophyte. So you can see it's surrounded by the nucellus tissue, and now this is the female gametophyte. Okay, so this female gametophyte will have, okay, 2 or more, okay, female sex organs called archegonium. So these are the archegonia. So are the female sex organs. Okay, so this functional megaspore, it developed into the female gametophyte. It developed into female gametophyte. So you can see here inside the female gametophyte we find 2 or more archegonia, or the female sex organs. So here this is how we see in case of the the megaspores, or in the case of female cones, we find the megasporophylls with megasporangia, that is ovule, and inside the ovule, so this is what we can see here. So megaspore mother cells, okay, will be one of the cell of the nucellus, and it undergoes meiosis and results in formation of 4 megaspores. Okay, out of that 3 degenerate, 1 remains, and that will develop into a female gametophyte. Okay, so these female gametophytes will not be released, and they remain, okay, inside the ovules, which are present on the megasporophylls. Now the pollen grains, which are the male gametophytes, so they are released, and these pollen grains are usually winged. So this is a pollen grain. So this pollen grain carries, so it carries male gametes. So it carries the male gametes, and here pollination will be by, okay, so pollination is by, okay, air. Okay, so air will be carrying these pollen grains from one plant to another plant. Okay, and these pollen grains will finally come and settle on the ovule. Okay, on the ovule, of course, this is covered by integuments. Ovule is covered by integument. Okay, and here is the female gametophyte, and this has, okay, the archegonia, the female sex organs. Okay, and so once the pollen grains they settle here, okay, so they are going to, so you can see the pollen grains settle and they are going to produce a pollen tube that carries the male gametes. So this is the pollen tube, and this is, okay, the pollen grain. Okay, so this is the micropyle of ovule. Okay, so you can see the pollen tube carrying the male gamete to the female sex organ. So these are archegonia. We know the archegonia contains egg cell. So it contains egg cell. Okay, so thereby resulting in fertilization. Okay, the pollen tube which will carry the male gametes into the archegonia, where it will get it will be fertilizing the egg cell. And after fertilization, okay, after fertilization, it results in the formation of zygote. So that will be 2n, and this zygote, okay, will become embryo, and by the time the ovule will develop into seed. So hence the seed contains embryo. So the seed contains embryo. Okay, so this is how the reproduction takes place in case of gymnosperms, right? So that's all about gymnosperms, right? Now we will see the next group of plants, angiosperms. Okay, so here out of all the groups of plants, these are the only plants that produce flowers. So these are flowering plants. So if you see a plant, okay, producing flowers, obviously it is an angiospermic plant, right? And so here the pollen grains, in case of reproduction, the pollen grains and ovules will develop in the flower. So they are going to develop in the flower. So here the flower have whorls. Okay, on the whorls we know, so this is the, okay, pistil, and these are stamens. So the stamens are the male reproductive structures. Okay, so these are stamens and this is pistil. Okay, they are the organs, the male and female, okay, sex organs. They have, so when we see in case of stamens, the stamens will show two parts. It has a filament and anther, and inside the anther, so if we take a transverse section, so we will see inside the anther there will be pollen sacs, and inside that there will be development of pollen grains. So these are the pollen grains, the male gametophytes. And coming to, okay, the pistil. So in case of pistil, we find three parts. The basal swollen part is ovary, and it has a long, okay, style, and on the top it has stigma. So stigma, style, and ovary. So inside the ovary we find the ovules. Okay, we know after fertilization this ovary is going to develop into fruit and ovules will develop into seed. and the seeds are covered by ovary. So and hence in this case of plants, that is angiosperms, the seeds are not naked, right? So here the pollination in this case will be will be by air. So it may be by air, water, right, or by animals. Okay, so finally the pollination brings the pollen grains onto the stigma. Okay, so here now the pollen grain germinates here, and this inside the pollen grain we know there are male gametes. Okay, so germination and okay, so here, so after germination we see that the pollen, one tube carrying the male gametes into, so here this is the pollen tube, male gametes into the embryo sac present inside the ovule. Okay, so this is the pollen tube. Okay, so now we'll see inside the ovule. Okay, inside the ovule, here also we will see the same process in the nucellus tissue. So one of the cell gets differentiated to form the megaspore, mother cell. So megaspore mother cell, which will be diploid, and this one will undergo meiosis and produces 4 megaspores. Out of that 4 megaspores, 3 degenerate and only one will become functional megaspore. So this functional megaspore will develop into embryo sac. So that will develop into embryo sac. So this is how we see the ovule, and it is covered by integuments. And we can see here it has micropyle. So this is an opening, the micropyle, and inside this is the embryo sac. So this embryo sac will show a few cells. So here you can see, and these 2 cells are synergid cells, and here is the egg cell. Okay, so we know these are 3 antipodals, and these 2 are the polar nuclei. So there will be 2 polar nuclei. So these 2 are synergid cells, and this is a egg cell. Okay, so here this is the pollen tube carrying the gametes, and then it passes through these synergids. So here in the synergids, the pollen tube breaks open. The pollen tube breaks open and then releases the male gametes. Okay, so one of the male gamete will fuse with the egg, the other one with the secondary nucleus. So here in this case, the 2 polar nuclei fuse to form secondary nucleus. The 2 polar nuclei fuse to form secondary nucleus, and the secondary nucleus, that will be diploid. So this is diploid. So in this case each polar nucleus is haploid. Okay, so here we will see a unique pattern of fertilization that nowhere in the plant kingdom is seen. So it's called double fertilization. So we'll see in case of angiosperms, double fertilization. So it is unique to angiosperms. Nowhere else in the plant kingdom is this double fertilization taking place. So this includes 2 processes, syngamy and the triple fusion. So it takes place with of course, with the two processes, syngamy and triple fusion. So here I told you one male gamete plus one egg cell. So one male gamete, one egg cell. So this will be syngamy, right? And the secondary nucleus that will be 2n, okay, plus one male gamete. So this is n. So here male gamete n and n. Of course, the syngamy results in the formation of zygote, that will be 2n. And secondary nucleus, okay, 2n plus one male gamete, and this is what we call the triple fusion because three nuclei are involved, okay. And this results in the formation of primary endosperm nucleus, okay. Of course, that later forms into endosperm, which will be triploid. So zygote will be diploid and endosperm will be triploid. So this endosperm is, okay, food to the developing embryo, okay. So the zygote will later become embryo, okay, by using this endosperm, and finally okay, of course, that's what it will become embryo, and the ovule by the time will develop into seed. So the seed will contain embryo, and this embryo may have one cotyledon or two cotyledons. If it has one cotyledon, then it is called monocotyledon, okay, or monocots. Then if it has two cotyledons, it is called dicotyledon plants, okay. So here in this case, the tallest angiospermic plants will be like eucalyptus. So this grows around, okay, 100 meters, and the smallest will be, the smallest plant will be Ulfia. Okay, it's Ulfia, so tiny that you can put it on your index finger, so small, so you can put it on the tip of your index finger. So that small is this plant, okay, Ulfia, right? So that's all about the general characters of angiosperms. So with this, I will end today's session. In the next session, we will begin with the objective on this plant kingdom.